Watson and Crick. So later in 1953, Mr. Watson and Crick, Francis F. Crick, James D. Watson, and Francis F. Crick, they have confirmed that the DNA is a double helical structure, a double helix structure. Which structure? A double helix structure. So when they confirmed it in their 1953, from the data taken from Rosalind Franklin and Maurice Wilkins, they got the structure of DNA accurately. Further studying the basic available from Rosalind Franklin and Maurice Wilkins. So how they found out the structure? Let's see. They confirmly studied the structure and said that it is having three parts. The DNA structure is having three parts. The first part was deoxyribose sugar. The first part was deoxyribose sugar. Second part was nitrogenous bases. Second part was nitrogenous bases. Nitrogenous bases included adenine or guanine or thymine or cytosine, a single repeating unit. One nitrogenous base in a single repeating unit of DNA. Okay. Then the third part, they have discovered that it was a phosphate group. It was a phosphate group. The phosphate group was like H3PO4. So each repeating unit of DNA contains one deoxyribose sugar, one nitrogenous base out of adenine A, guanine G, thymine T or cytosine C. One nitrogenous base is there in one kind of nucleotide. Nucleotide is a repeating unit of DNA. So they discovered these things. Now let's see how they appear. Deoxyribose sugar is a pentose sugar. Deoxyribose sugar is a pentose sugar. The deoxyribose sugar. Let's see the deoxyribose sugar. Deoxyribose sugar is a pentose sugar with a ring oxygen inside. With a ring oxygen inside. It has no oxygen at second carbon. It has no oxygen at second carbon. So its another name is 2-deoxyribose. It is also known as 2-deoxyribose. HOH HCH2OH. The fifth carbon is outside the ring. 4 carbons are inside the ring. 1, 2, 3, 4. So this is the structure of deoxyribose sugar. It is also known as a pentose sugar. It is also known as a pentose sugar. So this is the structure of deoxyribose sugar. Now let's see about the nitrogenous bases. How many nitrogenous bases are there? There are 4 nitrogenous bases. Adenine, denoted as A, guanine, denoted as G, thymine, denoted as T, and cytosine, denoted as C. These are 4 nitrogenous bases. These are 4 nitrogenous bases. Which bases? Nitrogenous bases. These are nitrogenous bases. Out of these nitrogenous bases, two are double ring structures known as purines, and two are single ring structures known as pyrimidines, So what are these? Adenine, guanine, these are double ring structure nitrogenous bases known as purines, and thymine and cytosine are single ring structures, T and C denoted as T and C, known as pyrimidines. Now let's focus on to the phosphate group. We will see the structures of all these four nitrogenous bases later in detail. First, let's focus on how the phosphate group appears. Okay, the phosphate group is in the form of H3PO4. Phosphate group is the third part of a repeating unit in DNA known as nucleotide. Phosphate group is the third part of repeating unit in DNA. So it is denoted as central atom P with 3 OH group, H3PO4. One oxygen is double bonded, another oxygens are with hydrogen singly bonded with phosphorus. Okay, so it is known as phosphoric acid. Generally these H are absent and the phosphate group carries negative charge, because of this phosphate group negative charge, the DNA has negative charge and it is hydrophobic in nature, also. Okay, so let's see the basic structure of DNA, the double helix, how it appears. Okay, so we will see the structure in detail now. The structure of DNA, it is like this: double helix model structure of DNA. As you can see, the DNA is having structure like this. You can see it clearly. So one end contains 5' group and one end contains 3' group. So at 5' end, fifth carbon, it is phosphate group there. I denote it as P circled by. Okay, and at 3' end there is hydroxyl group present. Both strands of DNA run in opposite direction. Likewise, the strands of DNA run in opposite direction. If there is P, then another P will be on the downside. Okay, from of another strand. There are bonds formed inside due to the complementary base pairs. So let's see how many bases are there. What is the structure of this DNA? Let's reveal this. Okay, 5' phosphate C. It is anti-parallel. If one end is 3', another must be 5'. If one is 5', another must be 3' OH. Okay. So each turn of DNA, this whole turn, these two twists makes one turn. It is 34 angstrom in length. One turn of DNA is 34 angstrom in length. The width of DNA, this width is only 20 angstrom. This width is only 20 angstrom units. There are 10 base pairs per turn. This is a complete turn having 10 base pairs. So 10 base pairs occupy 34 angstrom. Means one base pair occupies 3.4 angstrom of space. One base pair occupies 3.4 angstrom of space. Now there are major and minor grooves. This groove is major one. The major groove is approximately 22 angstrom in deepness and the minor groove is 12 angstrom in deepness. The minor groove is 12 angstrom in deepness. So this is double helical structure of DNA. It looks like a twisted ladder having 10 base pairs per turn, having 2 ends for each strand, 5 prime to 3 prime. Both strands are in opposite direction, means if one strand has 5 prime end on one side, then on the opposite there must be 3 prime end of other strand. These are bonded with complementary bases. So how bonding occurs? Let's see. If there is adenine on one strand, there must be thymine on the other strand. These two get bonded together with the help of two hydrogen bonds. A bonds with T with the help of two hydrogen bonds. A bonds with T with the help of two hydrogen bonds. Means A is a pyrimidine or purine. Let's say A is a purine beta. It is double ring structure. A is a purine. Purine bonds always with pyrimidine, or pyrimidine bonds always with purine. PY-PU or PU-PY pairing should be there. So you can write T can pair with A also with the help of two hydrogen bonds. It is not a covalent bond. It is a hydrogen bond. So what is the bond length? If you want to know, there is a bond length in AT or TA bond. Adenine bonds with thymine having the upper bond length 2.8 angstroms and lower bond length 3 angstroms. Opposite to this is second pairing, that is known as guanine to cytosine pairing. I will show it here. Guanine pairs with the help of 3 hydrogen bonds. How many? 3 hydrogen bonds with cytosine. Each bond is having different bond length. I will show the bond length here. The uppermost bond length of GC bond is 2.8 angstrom. Middle bond is having 3 angstrom bond length, and the lower bond is having 2.8 angstrom of bond length. G pairs with C with the help of 3 hydrogen bonds having the upper bond length 2.8 angstrom, middle bond length 3 angstrom, and the lower bond length 2.8 angstrom. All these are hydrogen bonds. All these are hydrogen bonds. There is a pairing which is present in DNA always in the form purine to pyrimidine pairing or pyrimidine to purine pairing. There is no purine or pyrimidine pairing, because it will distort the structure of DNA, double helix structure of DNA. So let's see about the nitrogenous bases now. How the nitrogenous bases work?